Okay, so welcome to this next video in which we are discussing the pathogenesis of rheumatoid arthritis. Okay, so we've just been discussing the pre-articular phase, the portion that doesn't actually involve the joints, okay? And in the pre-articular phase, what happens is that you get a failure of immunological tolerance, which leads to the production of antibodies which are directed against proteins which are citrullinated. Okay, now I want to stress that a antibody will bind to a specific citrullinated protein, okay? However, you don't just produce a single antibody which is against a single citrullinated protein. You produce many different types of citrullinated protein antibodies, basically. So, there are a whole host of citrullinated proteins that you start presenting to the adaptive immune system and start launching humoral adaptive immune responses against. So this is a non-specific response, basically. You're getting the humoral adaptive immune response initiated for loads and loads of different citrullinated proteins, okay? So there are many different citrullinated proteins. So let's say this is a citrullinated protein, protein A. Let's say this is a citrullinated protein, protein B. Let's say this is a citrullinated protein, protein C. Okay, I'll just put that there. Okay, so basically you're producing different antibodies which are binding to them. Okay, so you lose your tolerance to citrullinated proteins, basically, and you start producing different antibodies that are against these different citrullinated proteins. Okay, so these antibodies that are against citrullinated proteins are collectively all called anti-citrullinated protein antibodies. However, you should realize that these are different antibodies and they've resulted from separate adaptive immune responses, basically. Okay, so this is the anti-citrullinated protein antibody that is specifically against citrullinated protein A, and this is the anti-citrullinated protein B antibody, and this is the anti-citrullinated protein C antibody. Okay, so you start producing many of these anti-citrullinated protein antibodies, okay, and this can happen years and years before you ever get synovitis and rheumatoid arthritis. Okay, right, so what is now going to happen is that you're going to get the transition phase, right, and we've discussed that when you produce these anti-citrullinated protein antibodies, you might just produce an antibody, an anti-citrullinated protein antibody, which is against a citrullinated protein which is found within synovial joints. So let's say citrullinated protein C was found in the synovial joints, and you've produced an antibody against it, which is circulating within the blood. So you're poised and ready to get rheumatoid arthritis. However, at the moment you haven't got rheumatoid arthritis because citrullinated protein C is within the synovium, or potentially within the synovial fluid, whereas this anti-citrullinated protein C antibody is within the blood, so they can't get access to one another. So you haven't yet triggered the whole thing off, basically, but you're poised and ready. Okay, now what's going to happen is the transition phase. Okay, so firstly, a step that you might not have expected is going to occur. So what is usually found is that you start producing a new anti-citrullinated protein antibody. So very, very shortly before you actually develop rheumatoid arthritis, you start producing a new anti-citrullinated protein antibody. So an antibody which is going to bind to a citrullinated protein. Now, which citrullinated protein does it bind to? Well, it's specifically going to bind to a protein known as citrullinated fibrinogen. Okay, so I'll put this here. So this protein is going to be citrullinated fibrinogen. So what is citrullinated fibrinogen? Well, fibrinogen is a component of the blood. It's involved in the coagulation cascade. It's also known by factor 1 It is coagulation factor 1. It is essential within the coagulation cascade. It's what's converted to fibrin, and then fibrin is assembled into fibrin strands, and that's the process of coagulation, where you build this fibrin meshwork, okay, which either traps platelets and sews them together to make a secondary hemostatic plug, or it's designed to trap pathogens in and stop them from spreading, okay. So you can also get citrullinated fibrinogen, which has had some of its arginine residues citrullinated and therefore potentially has changed shape. And now we start producing an antibody which specifically binds to this anti-citrullinated fibrinogen, basically. So it's going to bind to citrullinated fibrinogen. So this is an anti-citrullinated fibrinogen antibody. Okay, so shortly before developing rheumatoid arthritis, what's going to happen is that you're going to start producing these anti-citrullinated fibrinogen antibodies. Now, citrullinated fibrinogen, like the normal fibrinogen, is circulating within your bloodstream, okay? So you're now chucking out anti-citrullinated fibrinogen antibody into the bloodstream where citrullinated fibrinogen is present, okay? So what's going to happen? Well, the two are going to bind together. So what you're going to end up with is immune complexes within the blood. So this is the starting point, basically. You're going to end up with immune complexes within the blood. Okay, what then has to happen? The next portion of the transition phase is that you're going to get some sort of damage to your joint, okay? So you're going to get damage to that synovial joint. So potentially it could be microbial insult, okay? So maybe you get an infection within the joint, okay? Or it could just be mechanical insult, okay? So you could just mechanically knock it and damage it, okay? But something that's going to allow a little bit of an inflammatory response into the synovial joint, okay? So you do need some sort of inflammatory response, okay? So if you get a microbial insult or a mechanical insult, so maybe just a little infection within the synovial joint, or just a little mechanical damage, basically, what that is going to lead to is a little bit of an inflammatory response in the synovium. Okay, and when you get the inflammatory response, what's going to happen is that vasopermeability is going to go up. Okay, so what's going to happen is that the endothelial cells which line the capillaries and the endothelial cells which line the postcapillary venules here, they're going to retract their outer boundaries, basically, and open up holes between them. And now what can happen is some of these immune complexes can come out of the blood and into the interstitial fluid of the synovium, basically. And now these immune complexes are going to activate a much bigger inflammatory response. And this is important to understand, okay? So initially, what happens is that you get a very small acute inflammatory response because of the mechanical insult, potentially, okay? And now you're going to get a positive feedback of this. So you're going to get these immune complexes coming in from the blood, where you've got this citrullinated fibrinogen bound to the anti-citrullinated fibrinogen antibody, and these are going to come and bind to sentinel cells in the synovium which have the Fc gamma receptors, okay? So basically there are three major types of sentinel cell within the synovium. Two of them we've drawn, okay? There's a third one that will be present, which I haven't drawn on here, okay? So we've seen macrophages, we've seen mast cells, but there's another third type. So let's now discuss these sentinel cells. Okay, so let me draw the third type first. So another cell type that you've got positioned in all tissues of the body, basically. It's constitutively present in all tissues of the body, and its job is in immune surveillance, basically. It sits and watches and waits, basically. Okay, and it's called a dendritic cell, and it's because it has these dendrite processes coming off the main cell body. Okay, so this is a dendritic cell. Now, although they do look like the cell body of a neuron, they have absolutely no relation to neurons. They're not capable of conducting action potentials. They will have an electrical potential difference across their cell membrane, all cells do, but they won't be able to conduct action potentials, because in order to conduct an action potential, you need certain voltage-gated ion channels, which they just won't express, basically. So they're not an excitable cell. Okay, so this is a dendritic cell. We've also seen macrophages, so I'll draw a macrophage here. So here is a macrophage, and we've said that macrophages have very prominent mitochondria. So I might draw a mitochondria here. Okay, there's a mitochondria. Okay, so this is a macrophage, and you do just have some resident macrophages in the subintima of the synovium. Okay, and their job is again in immune surveillance. They're always on the lookout for invading pathogens. Okay, and basically you then also have these mast cells, and again these are a sentinel cell. They're looking out for pathogens. Okay, right, and these mast cells are armed and ready with vesicles full of histamine. Okay, so let me colour this in turquoise. So this is histamine in these vesicles, and these are called histamine granules because they're full of histamine. Okay, right, so here is the mast cell. So these free cells are what are known as the sentinel cells. They stand and watch and wait for pathogens, and if they see pathogens, they will launch an acute inflammatory response. They'll send out alarm signals, basically. Right, so what's happened is that you've got a little bit of increase in vasopermeability because you've suffered some sort of microbial insult or mechanical insult. Now what's going to happen is these immune complexes are going to come in, okay, into the interstitial fluid of the synovium, basically. So here is the anti-citrullinated fibrinogen antibody, and here is the citrullinated fibrinogen. So this is an immune complex. So when you have an antibody bound to its antigen, this is called an immune complex. Okay, now all of these sentinel cells have on their surface a receptor. Okay, well, a receptor type, because it's not just one receptor, but have a receptor type which is specific for IgE, and the normal antibody, I should say this actually, the normal antibody that you produce is going to be IgG rather. Sorry, did I just say IgE? I meant IgG. Sorry. So, start again. Right. The normal antibody that you produce in a humoral adaptive immune response is you produce IgG antibodies, one of these four types here. Okay. Now, let's say that this anti-citrullinated fibrinogen antibody is of the IgG type. Basically, these sentinel cells have receptors which bind to the Fc region of IgG antibodies. So it's the Fc gamma, because you've got the fragment crystallizable of a immunoglobulin G molecule, which, remember, the analogous Greek letter for that was gamma, and it's a receptor, so it's an Fc gamma R, basically. Now, there are many different types of Fc gamma Rs, but we'll just keep this simple and refer to them as Fc gamma Rs. So these are receptors which will bind IgG. Now, why? Why do these sentinel cells have receptors for IgG on their surface? Well, the idea is that you will be able to mount an IgG molecule on this Fc gamma R molecule. So here is our IgG molecule mounted, and basically now, remember, what is the purpose of producing IgG? The purpose is that you've produced this IgG against a protein that should be associated with some terrible pathogen, okay? So, Now, once the IgG is bound on the surface of the sentinel cells, basically you are giving the sentinel cell new tools. You are giving it new tools for the detection of pathogens, because if that incredibly dangerous pathogen ever comes back and it has the protein to which this antibody, this IgG molecule, is targeted on its surface, then, or even better, it's secreting it, then basically that protein will bind to the IgG molecule. That will activate the Fc gamma R molecule on the surface of the sentinel cell, and that will then activate the sentinel cell. So it's a way of detecting pathogens. It's a improvement, basically. It's a way of improving these sentinel cells' ability to detect pathogens. If they've got these IgG molecules bound to them, then they can now recognize new proteins that they weren't capable of recognizing before. Okay, so now we're bringing in a ready-made immune complex. We're bringing in an IgG molecule with its antigen bound, and we're going to stick this on our Fc gamma receptors on the surface of our free sentinel cells within the synovium. And what's going to happen? Well, they're going to go wild, basically. They're going to go, oh my goodness, we found the antigen of some incredibly dangerous pathogen. We need to start sending out alarm signals. And indeed, they do start sending out alarm signals. So, the dendritic cells and macrophages start producing interleukin-1, okay, IL-1 for short. And this is so important, I should probably write its full name out. So this is interleukin-1. And they also start producing tumor necrosis factor alpha, which is often abbreviated to TNF-alpha. So this stands for tumor necrosis factor alpha. And TNF-alpha is often just abbreviated to TNF, so it's often just called tumor necrosis factor. And the reason for that is that although there are different tumor necrosis factors, for instance there's tumor necrosis factor beta, there's also tumor necrosis factor C, tumor necrosis factor alpha is by far the most important of the tumor necrosis factors. Okay, so you know, if you see tumor necrosis factor without a clarification at the end, if you see it with nothing on the end, then you can assume that people are talking about tumor necrosis factor alpha, okay, because it's the main one. Now, mast cells, they secrete a different, well, they do secrete these also to an extent, but the main one they're going to do is they're going to release histamine. So they're going to degranulate, and they're going to release all the histamine from their histamine granules. Okay, so these alarm signals are going up in the synovial fluid. Okay, so you've got interleukin-1 being poured out into the interstitial fluid of the synovium, and you've also got tumor necrosis factor alpha being poured out into the interstitial fluid of the synovium, and you've also got histamine being poured out into the interstitial fluid of the synovium. Some of this will end up in the synovial fluid as well. Now, what are these going to trigger? Well, they're going to trigger the acute inflammatory response. Okay, so these are going to act on the endothelial cells of the blood vessels within the synovium. Okay, so remember, the blood vessels within the synovium were the terminal arterioles, the capillaries, and the postcapillary venules. So let's draw a picture of this down here. Okay, so we have a terminal arteriole here. Okay, and this will divide up into capillaries. So I'll draw a little picture like this. Now this isn't specifically in the synovial membrane, because of course the capillaries should be in a different layer to the terminal arteriole, but this is just to get the point across. Okay, so here are the capillaries, and they'll then reconverge to form a postcapillary venule, which I'll draw here. So this is the terminal arteriole here. Terminal arteriole. Okay, and then it branches into these capillaries here, and then finally the capillaries reconverge into a post-capillary venule. Okay, so let's see what effect these mediators are going to have on the terminal arterioles, the capillaries, and the post-capillary venule. Okay, so histamine is going to act quickest. The mast cells will degranulate and release histamine, and histamine is going to go to the endothelial cells, and it's going to cause type 1 activation. Now, of the endothelial cells. Okay, now histamine is both released extremely quickly and it also acts extremely quickly. So degranulation occurs extremely quickly, and then the actual speed that it causes type 1 activation of the endothelial cells is also extremely quick, basically. Interleukin 1 and TNF-alpha, these are both proteins. These have to be produced by the dendritic cells and macrophages, and also when they go to the endothelial cells, they cause something called type 2 activation. And if you want to know the actual pathways of type 1 activation and type 2 activation, I have entire videos on type 1 activation and type 2 activation respectively in my inflammation and angiogenesis playlist. Okay, what we're going to study is the big picture. Okay, so type 2 activation, not only do the mediators of it not only are the mediators of it produced slower, okay, so not only do they take actually longer to produce, type 2 activation, once they've actually got to the endothelial cells and they're binding to the endothelial cells, the actual process of the endothelial cell undergoing type 2 activation again takes much longer because it involves changes in protein synthesis, okay, so this one takes hours, this one takes minutes, basically. Alright, but what are the overall results of both of these? Because pretty much they're very, very similar. Okay, so what they're going to trigger, okay, is the acute inflammatory response. Okay, so acute inflammatory response. Now, what are the key components of the acute inflammatory response? Well, the first is that you're going to get vasodilatation of the terminal arterioles, basically. Okay, so the smooth muscle cells which line the terminal arterioles, those are going to relax, and that means that the rings of smooth muscle cells are going to dilatate, and this is going to cause dilatation of the lumen of the terminal arteriole, which is going to cause an increased blood flow to the synovium. Okay, so you're going to get vasodilatation, which is going to lead to increased blood flow to the affected area. Okay, so vasodilatation, and this leads to blood flow going up. Alright, now why is that helpful? Well, actually it's not helpful at all in this case, but why would that be helpful if we actually had a pathogen? Well, because if we're initiating this response, which is exactly what you would initiate if you had some incredibly deadly pathogen present, and that's what the sentinel cells think is happening. They've got this immunoglobulin G molecule with an antigen bound to it. They don't know it's an autoantigen. They think that this is some protein associated with some horrible pathogen, and they think they're therefore being heroes in initiating this acute inflammatory response. So you're initiating the same response as you would for a pathogen. So why is this helpful for a pathogen? Well, because if you have a pathogen present, you need to bring in troops that are going to help kill or at least contain the pathogen. Okay, and all of these troops are within the blood, basically. So we need to get them out of the blood. So if you vasodilatate the terminal arterioles, which are leading to the capillaries and the post-capillary venules, you're going to get more blood flowing through the affected area, and therefore you've got a greater number of troops, basically, that you could possibly recruit from the blood into the interstitial fluid. So that's why vasodilatation leading to increased blood flow is actually helpful. Okay, right. Then you also get increased vascular permeability. Okay, so increase in vascular permeability. Increased vascular permeability. Right, so what's basically going to happen is that the endothelial cells are going to retract their outermost portions, basically. So if I just draw this, so basically if you look at the capillaries and the post-capillary venules, you have endothelial cells like this. Okay, so this is in non-activated endothelial cells. So you have no gaps basically between the endothelial cells. What's going to happen is that we're going to retract these outer edges basically like so. Okay, at least in well, type 1 and type 2 activation will lead to this. Type 1 activation will lead to the pulling back of them, basically. It will lead to the contraction of the endothelial cells, whereas type 2 activation leads to the permanent dismantling of this outer portion of the type 2 endothelial cell. So the endothelial cell, sorry, type 2 activated endothelial cell. So the endothelial cell actually changes shape after type 2 activation. And this means that you're going to have much bigger gaps between neighbouring endothelial cells. So fluid from the blood can move through these gaps in the endothelium. They can move through these holes into the interstitial fluid, basically. So increased vascular permeability. And the final thing that's going to happen is you're also going to start recruiting leukocytes. Okay, so leukocyte is the fancy word for a white blood cell. Okay, so leukocytes are not big enough to fit through these holes in the endothelial cells, at least not without help. So the endothelial cells are going to help the leukocytes through. Now, you might ask, well, what was the purpose of the increased vascular permeability if you're going to have to do other things to get the leukocytes across? Well, this is going to allow fluid from the blood to move into the affected area, okay, and this is going to bring many different proteins with it, which are going to help fight troops, or in this case cause problems, because we haven't actually got any infection. We've got you know, a perfectly healthy synovial joint. Okay, so we'll continue this discussion where we'll discuss what happens next in more detail.